Welcome, my dear students. Hope you are doing well. So, regarding the plant biotechnology and physiology courses, BBT C202, module 5, I think. Yeah, it's module 5. The molecular breeding and marker assisted breeding is already taught by, I think, Sayantan sir. STG sir is already taken the class and provided the note, I think. So as you requested, so few portion you are not able to understand. So I would like to discuss about these few portions regarding marker assisted selection and molecular breeding actually. Okay. So thing is that first of all you need to know actually what is a marker assisted breeding. So in the term you can see marker assisted breeding or marker assisted selection, whatever you may say. MAS, S for selection, MAB, B for breeding. So MAS or MAB, marker assisted selection or marker assisted breeding. It is the thing using molecular marker how you can improve the crop. That crop may be a plant crop or animal crop or any microorganisms strain development or regarding their phenotypic generation, new variety release, etc., etc. So thing is that using markers. So you are aware about that already. The DNA marker means marker is like a bait. Okay, bait. When you want to catch the fish from a pond, so you use a bait in the fishing rod so that the fishes can attract. Okay, and you catch the fish. So fishes are the genes, and pond is you consider the whole pond is like a genome, and all the fishes are like the genes, and your fishing rod in the bait, the bait is the molecular marker that you are targeting. Suppose for katla you will use different bait, for puti mach or for geol fishes or for some different fishes, so you use different molecular markers, different bait. So likewise here also there are several baits or like the molecular marker like RAPD. I have already discussed about during protoplast fusion characterization, hybrid characterization. Molecular marker like RAPD, ISSR, SSR, and RFLP, AFLP. So these are the markers. Okay. So markers how they help actually. So markers means targeting. Suppose your target is to develop a new crop variety. So when you are going to develop a new crop variety, so your target should be in terms of maybe your crop variety will be high productive or disease resistant or maybe pest resistant or maybe environmental drought resistant, cold resistance, all the things will be there. Okay, so accordingly you have to choose the marker, your bait. Okay, so here is the cycle how actually marker assisted selection is carried out. So what is the step by step? Okay, so to achieve your goal, so here you can see, so first is the marker assisted selection is the population development. Suppose you want to work with rice, you want to develop a disease resistant rice or paddy. Okay, rice or paddy or wheat. Okay, let us consider for rice plant. So you want to develop a high yielding rice plant. So develop a plant grown in the directly field, that is nothing but the population development. So in the field phenotyping by morphological observation, you just pick, you just see which plant is giving high yield. Okay, so that particular yield character, this is the trait or character. Okay, so this trait you want to insert in your next generation, that is your motto. So high yielding variety or high yielding plant you are selecting. So population development, so in the field, then phenotyping means morphological observation, genotyping means genetic observation, with working DNA, RNA. Genotyping. Phenotyping means the phenotype is the morphological observation that you can see in your naked eye, visual observation. So you can see the high yielding productive variety. Then high yielding productive variety, you just choose, select. Okay. Then next step is genotyping. So once phenotyping is done, phenotyping is means you choose already the high yielding variety. Then you bring it into the lab and then extract the genome or DNA and then use different molecular marker like RAPD, ISSR, SSR for genotyping. Here is the genotyping. Okay, gene. You're working when you're working with gene, then it will be termed as genotyping. So once genotyping is doing so, oh, okay. So once the phenotyping is done, then it will be brought to the lab for genotyping analysis. Okay. So your target is to catch the gene is responsible for high yielding. So high yielding is terms of production of more glucose or production or the synthesis of the carbohydrate polysaccharide, because you are working with the rice plant. So that means the photosynthetic gene is responsible. That gene is your target. So that if you are, or carbohydrate synthesis gene, so there are many genes is related to high production. So if you are able to pick up or identify the gene once using the molecular marker or like the bait, means you are targeting to catch the fishes. Okay, so once it is done, so you identify it, then you just make a map. Okay, you just develop a map. That map is nothing but QTL. QTL is the quantitative trait locus. Is a locus that correlates with variation of the quantitative trait in the phenotype of population of organism. So once your genotyping is done, then you just compare with some positive control and negative control. You pick the high yielding variety and you just take a normal average variety and less variety. So this is the three varieties you are choosing, three plants you are choosing. So once DNA fingerprinting is done, that means genotyping using molecular marker, you just make a map. Based on the map, you just choose your target prominent band. So I'll show you in the next slide how QTL mapping is done. So that means it is nothing but the comparative study. QTL mapping is nothing but the comparative study. Don't confuse. Quantitative trait loci. Quantity means the quality you are judging among the population. Nothing more than that. So don't make it so complicated and confused. Then QTL validation. Validation means you are selecting which one is the best. Okay, so QTL validation means you are selecting which one is the best. Then marker validation. So once QTL validation, so you already choose the this variety is good product in terms of production, then you go for the marker validation. Okay, so my RAPD marker, this marker worked very well. I receive a good result, and then your marker assisted breeding is done, completely done. Okay, so this is the overall thing of marker assisted selection. So I hope this is enough for the overall step. So now, so this picture is little bit explaining, so you can identify. So here is suppose two plants are crossing. This is for the back cross. Okay, so leave it. So here, see. So how genotype phenotyping to genotyping is done. So this is very important, how genotyping to phenotyping is done. So conducting a mass program when tissue sampling, see here is the garden. So you are collecting the high yielding variety. Then DNA extraction, DNA you just brought into the lab and using the chemicals and all those things, you just extract the DNA, then do the PCR polymer chain reaction, then gel electrophoresis, gel means the agarose or polyacrylamide. Generally, we use for DNA agarose gel electrophoresis, then marker analysis. This is the marker validation. See, marker validation. So here there are lots of bands of individual plants. This is one lane, one individual plants. Okay, this one lane indicates one individual plants. So as per your target, you will pick a single band, which one is very unique. Which one is very unique, a single band you will pick. Okay, so based on this band, you just cut the band and go for sequence analysis. So sequence one, sequence analysis is done, so you will have the sequence, and you, that sequence, with the sequence you just blast means there is a NCBI gene bank in the USA. NCBI gene bank data, gene bank is the collection of genetic resources. So you just check your gene is matches with any other genes of any other particular crop or not.